Okay, so now I want to talk to you about flowering plants. And most of us are familiar with flowers, but it's useful and worthwhile to sort of explore the various parts and pieces of a flower and to understand sort of the homology of the floral organs and how they relate to the organs in gymnosperms and ferns and mosses. Okay, so here we have a longitudinal section of a flower. You see we have purple petals, we have the sepals, the leaflets that control that wrap up the bud. But we're more interested in sort of the internal organs or those that are sort of related to reproduction. And here we have the internal part are the pistil, Pistil, and the pistil is composed of the stigma, the style, and the ovary here. Okay, and if you take a look over here, you see we have the pistil, and then we have the stigma, style, and ovary. Now, the ovary is something that is new. It's not a new structure per se, it is a modified leaf, but it is a new organ. So it's, I guess, a new use of an old organ. And the job of the ovary, if you look inside the ovary, what you see is that you have the ovules. Now, remember, in the case of the gymnosperms, these ovules were just out in the open environment. Gymnosperms were naked seeds. They didn't have an ovary. They didn't, in other words, they didn't have a leaf that sort of wrapped around and sealed the ovules inside of an ovary. That's an important innovation for lots of reasons. A mature ovary is the fruit. So mature ovary ripens and turns into a fruit. So whereas the ovule ripens into the seed, the ovary ripens into the fruit. So we have ovaries and fruits in the angiosperms. Now they're still heterosporous. We have our anthers here. I'm not going to talk about the anthers. They're really interested. Our discussions have really focused on the megasporangia and now specifically the ovaries. Okay, so now what I've done here in this next image is I've taken a cross section, I've cut it just like this, and now we're looking right down inside of the ovary. So here we have two different ovaries of different species, something from the lobeolioids and something from the campanuloids. And you see this is just a sort of a part of the organ, the central part of the ovary or the fruit, and attached to that are all these ovules. Now these are all seeds. They were megasporangia. Remember that from the gymnosperms that ripened and that ripened into eventually we have contain the seeds. So here we have the ovules, here we have ovules, and probably seeds at this point. Okay, so now let's examine that a little more closely here. So here is a cross section of a lily ovary here, and I've made a very thin section, and now we're looking at this under a microscope. And I want to zoom in because each one of these is megasporangium or an ovule. Okay, and if you look at this one here, for example, you see that there is actually um, a megagametophyte, most likely inside of this megasporangium. Again, the megasporangium is eventually becomes part of the ovule. Okay, so now the reason why I want to talk about this is because this megasporangium here is actually homologous with the megasporangium in gymnosperms, and that is homologous to the sporangia in mosses and ferns. Okay, they're homologous. So there you see some of the similarities, I think, between the gymnosperms and angiosperms. Really what is new here in terms of structures is the ovary, or a leaf that sort of wraps around and conceals the ovules from the external environment. Okay, now I want to take a look at this megasporangium. Now, I don't expect you for this class to understand all the terms, antipodals, synergids, etc. But I do expect you just to understand what we're looking at. So this is the ovule or the megasporangium. And inside of the megasporangium, we had megaspores that germinated and gave rise to megagametophyte. And now, unlike the case with the gymnosperms, the megagametophyte of angiosperms develops through mitosis, but it doesn't get very far. In this case, we have 1, 2, 3, 4, 5, 6, 7 cells. So this particular gametophyte grows to 7 cells and then it stops. No further development happens unless it is fertilized. And the way the fertilization works in angiosperms is fascinating and quite interesting. So if we go back up to my flower, what happens is I'm going to draw a pretend pollen grain. A pollen grain lands here. That is not the size, okay? That pollen grain, then it lands there. It's brought there by an insect, possibly or the wind or however it gets there, but it lands there. And remember, the pollen grain is the male gametophyte. That pollen grain will actually germinate, and it will grow a tube down the style and into the ovary, and eventually getting into the megasporangium. Let me go down here and do it sort of a cartoon. So imagine, do it like a cartoon. Imagine I imagine that I have my pollen tube growing down. I'm inside of the ovary, okay? This is all inside of the ovary. Now the pollen tube grows down and it enters into the megasporangium, okay? And as it enters into the megasporangium, it comes in and it deposits one sperm here to fertilize the egg. The tube continues to grow and it grows into the middle here and stops here. And it drops off another sperm and fertilizes something very interesting, the polar nuclei. Now, if you look at this image, what you see is that we have a lot of cells in here with a single nucleus. All of them have a single nuclei. The one in the middle has two nuclei. We refer to these as the polar nuclei. So essentially, this is a gametophyte, so it's haploid as all other gametophytes, but it really has one diploid cell in the middle, and the polar nuclei are diploid, two nuclei. Slightly different than the diploid that we've talked about in terms of genetics, but it is diploid in terms of the material that's inside of the cell. So when the pollen fertilizes when the pollen comes in, the tube comes in, and the sperm fertilizes the polar nuclei, you wind up getting a tissue in here that is 3N or triploid. And that tissue begins to develop through mitosis to produce what we call the endosperm. And the endosperm begins to proliferate and grow, and it basically replaces all of the megagametophyte tissue in here, leaving just space enough for the fertilized egg. Let's just make the fertilized egg red. So the egg is here. That eventually goes to the zygote, becomes the embryo. And so the embryo now is embedded in this triploid endosperm tissue inside of the megasporangium. So the innovation here is fascinating because in the case of gymnosperms, they make large multicellular gametophytes whether they're fertilized or not. It's a net energy loss. In the case of the angiosperms, they only make an 8 or 9 or 10 or 12-celled gametophyte and they stop. If they are fertilized, then that second or that double fertilization event, the second fertilization event, creates an endosperm that then grows by mitosis to create the nutritive tissue that feeds the developing embryo. So we call that process in flowering plants double fertilization. That process gives rise to the endosperm. Here we have a nice image here, zoom in, of exactly that. So here is that central cell, and you see the polar nuclei with two nuclei inside one cell, and the egg is down here. Okay, so it's a fascinating look into how double fertilization works in flowering plants. And the final product, or the final bit, is the mature ovule, which is a seed. So here we have a seed of lily, and it looks kind of odd, but this is all part of the seed coat here. And inside the seed coat, you have the embryo, or the baby plant, and you have it embedded in a matrix of endosperm. So we have a diploid cell wall, or seed coat, a diploid embryo, and a triploid endosperm. Fascinating genetics that happens in the seeds of angiosperms that have double fertilization.